This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV. This Week in Virology, episode 1243, recorded on August 8th, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. And it is a gorgeous summer day here in Western Mass. Clear blue skies, 80 Fahrenheit, 27 C, light wind, just absolutely lovely. The only downside is we haven't had rain in a long time, and we're not expected to get rain anytime soon.
Here in Chelsea, it's also 27 C and sunny, not a cloud in the sky. It's going to be like that through Wednesday. How about that? Nice. Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to be here. As I'm sure no one will be surprised, it is the same here. Also 80 Fahrenheit, not a cloud in the sky, and just a lovely day.
Wow. And from Austin, Texas, Rich Condit.
Hi, everybody. Well, Alan said it was a gorgeous summer day where he was. I will tell you that it is a summer day here, which is by definition not gorgeous. It is clear blue skies and all of that, but it's 99 degrees.
Ooh.
And I get tired of that after a while.
But it's a dry, right?
It's a dry heat.
Dry-ish. Okay?
Got
but,
a little bit of humidity.
Over about 95 degrees, humid or dry, is not, a couple of days of it is okay, but, a couple of months of it gets tedious. It's not 110.
You like these programs where we give you the real science, no lies, no disinformation. You're going to hear some of that in a moment in our news section. We'd love your support to help get the word out, help us to make these programs. We don't put advertisements on these programs. We depend on your support. Go to microbe.tv/contribute. All right, we have two news items. Oh my gosh. First, yesterday, the latest executive order, which is a shorthand for trying to get around the law as president. This is called Improving Oversight of Federal Grant Making.
And just as a guideline here, that these executive orders coming out of the Trump regime are worded kind of like Pravda, where you can just read the opposite of whatever it claims to be, and that's what's going on. So, destroying oversight of federal grant making.
This is just ridiculous. I'm going to quote from Angie Rasmussen, who writes a column on Substack called Rasmussen Retorts. She writes, yesterday's executive order improving oversight of federal grant making is simultaneously some of the most vicious, depraved, and illegal BS to be issued in the Trump administration's war on science, but also a very stupid self-own for America. If enacted, it will devastate science, health, and the economy. It's a blatant theft of congressionally appropriated taxpayer funds and an attempt to further undermine democracy at the expense of America's global standing and the well-being of its people. So this executive order has purpose. Section 1 is purpose. Every tax dollar the government spends should improve American lives or advance American interests. Really? So the $1 billion to make a new 747 for the president is going to improve my life? Okay, going on. This often doesn't happen. Federal grants have funded drag shows in Ecuador, trained doctoral candidates in critical race theory, and developed transgender sexual education programs. This is not America if you don't want that. I'm sorry. In 2024, one study claimed that more than 1/4 of new NSF grants went to diversity, equity, and inclusion and other far-left initiatives.
They do know there's a section asking for that, right?
Yeah.
Yes, absolutely.
Broader
Both
impact?
NIH
So
and NSF.
the upshot of this executive order is that all grants will now have to be reviewed by political appointees,
Yep.
to ensure that they are in line with the prevailing ideology.
Yes.
Yep,
But
that's really what it does.
this
That's
far right nonsense, I don't know if that's allowed, right? Congress appropriates the money to NIH, and then NIH decides how to dole it out, right?
yeah.
I don't know. I don't know if you can modify that. I'm sure there'll be lawsuits.
And even if you didn't think about the ideology part, which is atrocious, why do we want a political appointee deciding if my grant is good?
They
What
don't know
do they
anything
know
about
about
it.
immunology?
They
They
don't know
don't.
anything about it.
They
Nothing.
don't.
They're going to look for keywords, right?
Yeah.
And then, HIV and COVID, they're going to say, oh, we don't need this anymore.
And the other thing is, I mean, it'll occur to people that waiting for all these political appointees to approve stuff is going to gum things up and delay grants. And that's also part of the point of this, to delay government expenditures so you can have more tax breaks for billionaires.
The one of the things that constantly annoys me is that a basic um, motivation or rationalization for this is to counteract a far left ideology, which is to me remarkably hypocritical.
Yep.
Because I don't think there really is an ideology associated with any of these things that they're fighting. It's just a natural democratic evolution of thinking in the country. And the hypocrisy is that what they're doing is instead injecting a far right ideology into the process. Okay, so this is the kind of hypocrisy that just drives me completely up the wall, and it's so anti-intellectual and basically anti-freedom that it just drives me nuts.
Yep.
So, one of the other things I think about with this is I think about how many people, as all of these changes are happening with grants in all sorts of different ways, how many people are going to potentially have trouble with keeping their employment? How many jobs are going to be lost because people don't have the ability to fund their lab with grants? So, how many labs will close? How many, technicians and other lab support staff are going to lose jobs? And it's a significant number. I saw something that was actually trying to get at that on social media, and I don't remember the exact details. I just did a quick Google search of what I remembered because the thing that I saw was talking about how many jobs are being impacted by some of these grant details, and I think they were only talking about maybe in the DC area or something like that. But they were comparing it to things like how many coal miners there are in the United States, who are, a group that we often have to think about when we think about making sure we have appropriate employment in other areas of politics. And my quick Google search said that there are 6.3 million people who do science in the United States and 43,000 coal miners.
Yeah.
And so I think that if people also should realize that many of these scientists who are losing out on their grant funding are also scientists who are employed in the United States. And all of these things are hitting some of those pieces that people don't think about as being ideology in any way, shape, or form. We are going to have more unemployment. We are going to have other issues because of this.
Well,
And there are follow-on impacts from that as well, because one lab is not just one person sitting alone in a room thinking. I mean, there are people in theoretical physics who do that, but they don't really have a whole lot of grant money coming in. These grants are funding laboratories full of other people and supplies and consumables, and they're participating in the economy. And this grant money is not sitting around under a mattress somewhere. It goes into the lab and it goes right back out into the economy. So, I mean, this is ultimately you could look at as an economic stimulus program.
Yeah. So it's sort of like, okay, so we're going to harm our knowledge
Mhm.
and we're going to harm all of these economic activities. I believe Angie called it a self-own, and that's a understatement.
Yeah.
Fundamentally is inhuman and doesn't make any sense. Makes zero sense whatsoever. This
Well,
is not what
we've been
most of
asking
America
we've been
wants.
asking for years, what's it going to take to wake people up? And I still don't have the answer to that. But so far, it hasn't been enough, and it's pretty bad. Okay, it's gonna take a crisis that I don't want to see, but unfortunately, I think that's where we're headed. A real crisis.
Well, speaking of crisis, our next news story, I have an announcement from the U.S. Department of Health and Human Services. HHS winds down mRNA vaccine development under BARDA, Biomedical Advanced Research and Development Authority. And RFK Jr. says, "We reviewed the science, listened to the experts, and acted. They're terminating 22 mRNA vaccine development investments because the data show these vaccines fail to protect effectively against upper respiratory infections like COVID and flu. We are shifting towards safer, broader vaccine platforms that remain effective even as viruses mutate." So, first of all, this is ridiculous because the mRNA vaccine saved millions of lives. They work. So he's lying by saying they don't work effectively. We do not have platforms that remain effective as viruses mutate. People are working on it. But apparently that's going to be at risk because of the previous story, NIH grants. So the justification for this is just a lie. And I have also an article from the Times by Apurva Mandili. Scientists disputed Mr. Kennedy's claims as inaccurate. If you don't believe us here today, okay, we got some other scientists. By issuing this wildly incorrect statement, the secretary is demonstrating his commitment to his long-held goal of sowing doubts about all vaccines, said Jennifer Nuzzo, director of the Pandemic Center at Brown University. Had we not used these life-saving mRNA vaccines to protect against severe illness, we would have had millions of more COVID deaths. All right, and one more thing before you guys can chime in. Here's Kennedy again. Let me be absolutely clear. HHS supports safe, effective vaccines for every American who wants them. That's why we're moving beyond the limitations of mRNA. This is a lie. You do not support safe and effective vaccines. You're canceling them left and right. He just lies. You know how he's lying? His lips move. Every word out of his mouth is a bloody lie. I cannot believe it. Okay, that's enough of my rant. What is it called? Racanielling.
Racanielling, yes.
So
But
I
yeah,
don't understand. I don't understand why he's picking on mRNA vaccines, except that I don't know. I guess it's kind of catchy or something like
Political.
that.
it's part of the right-wing conspiracy thing post-COVID.
Right.
The mRNA vaccines were going to reprogram our DNA. They were part of, they were right at the core of what people were making up stupid things about. And so they are a lightning rod for this, and they're an easy target.
Because
as a result.
this is absolutely incredible because this is the anti-vax camp is running the show.
Yes.
All right.
Yeah.
And
Yeah,
it's
it is.
it's lunacy. It's
Yep.
lunacy.
Totally. Total lunacy. I mean, taking COVID vaccines away from pregnant women and people under 65 is lunacy also. Pregnant women are at risk for severe COVID, and we know that younger people get severe COVID. They get long COVID as well, and vaccination prevents it.
So the PBS NewsHour ran a piece on this last week, and two features of that stick in my mind. One was a clip of Kennedy talking about this and announcing what he was going to do, and it was absolutely clear that he had absolutely no idea what he was talking about. And the guest on it that talked about the whole thing was Michael Osterholm, and he was frankly quite good. And sort of laid the whole thing out and also said, Kennedy doesn't have the slightest idea what he's talking about. He's completely clueless.
Yeah, I think that people can find information about kind of what a great innovation mRNA vaccines have been and how many ways they have really improved and have so much great potential. And so that's all out there. You can find that in a whole bunch of places. I just dropped in the show notes a link to a paper in JAMA that I remembered having seen. It came out on July 25th, so, a couple of weeks old. But it actually looks at the number of lives saved by COVID vaccination and does modeling. So if you'd like evidence that says that he is lying, that in fact, specifically evidence that says these vaccines, though it does not single out the mRNA vaccines, you can see this paper from JAMA basically happening at the same time as he's saying this, indicates 2.5 million deaths averted.
So Colbert has even roasted him on this. Colbert said, "Bad news for fans of living." Oh my gosh. Oh my gosh.
Well, here
And
comes the crisis.
Yep.
that's
Here
right,
it comes.
manufactured.
Yeah, it's
Uh,
economically, politically, health news, everything, everywhere you look. It's just one big crisis. It's unbelievable.
And he previously canceled the H5N1 mRNA vaccines. So next time we have a pandemic, hopefully he'll be long gone and we can do things normally. We shouldn't have one in the next 10 years, I would guess.
But careful there.
Yeah, you don't have
We can't
pandemics
really.
so
I mean, there's
close.
no guarantee just because we had one. It's
No.
like you could have a 100-year flood 3 years in a row.
Yeah, but we're not gonna I can't guarantee anything, but if you look at the trends,
Yes, right. It
we're
would
not
be
gonna have
surprising.
something like COVID didn't happen since 1918.
Yeah.
I am. I feel like all of this is not good karma to be saying.
Yeah, oh, right.
No, I think it's fine. We are scientists. We don't.
I just
deal
hope
with
I live
harm.
long enough to see all this turn around. I want
Yes.
to
I
see.
think we need to get rid of him and his ilk so that science can resume. But it's not going to be flipped back on with like a switch
No.
because
No,
the damage
no, we're
takes
going
a
to
long
have to
time
dig out of
repair.
this.
Okay, on to the science, which is much more uplifting, I should say. I wanted to talk for a while about Lenacapavir,
Yeah.
capsid inhibitor for HIV-1 treatment and prevention. This is amazing, this drug. Stay tuned. Don't drop off for a while, okay? Listen to this. I have a review article here because I wanted to cover the whole gamut of this drug. This is a paper in Biochemical Pharmacology. It's a review article by de Klerk, Zhang, Li, Dong, Kuri, Ruzek, Kroll, and Tan. They're from China, they're from Belgium, they're from Brazil, the Czech Republic, Poland. Yeah.
I think that's it.
No US.
No, the U.S.
So
is going away.
is going away. That's right. So Lenacapavir is the first capsid inhibitor for HIV-1. It binds. So HIV-1 is an enveloped virus. It's a retrovirus, an envelope with spikes in it, and then within it there's a capsid, and then within the capsid is the genome. And the capsid is made up of protein subunits called hexamers, and this drug binds between the subunits of these hexamers. It interferes with multiple steps in virus reproduction. So it interferes with assembly, and it interferes with nuclear import. So it's extremely potent, as you will see, and it lasts a long time. When given subcutaneously, you need 2 shots a year to prevent infection, prevent you from
A
getting
nice thing
AIDS.
about targeting the capsid of an envelope virus is that this envelope virus in particular, HIV, it can vary a lot. The proteins on the outside of the envelope can vary, and so it's hard to target with vaccines. The capsid is very well conserved. And so if you muck with the capsid, you mess up the structure of the virus, and it doesn't have a lot of evolutionary space to fix that problem. So it's a really promising approach.
Yeah, and the HIV capsid is sort of internal to the envelope. It contains the RNA genome as well as some of the enzymatic machinery for the virus. And there has been some debate in the field about some details of the capsid for a while. And in fact, some of the understanding about what's going on there is very relevant here because it has been shown that the capsid actually stays together throughout the cytoplasm and is sort of a nice shield for the genome. That's where reverse transcription happens, is sort of in this shielded capsid that's traveling through the cytoplasm. And then that capsid interacts with the nuclear pore to deliver the DNA directly into the nucleus. And a number of investigators have shown this really beautifully over the past few years. This started with some understanding of a host protein that bound to capsid that seemed to block HIV replication in certain situations, and sort of how in the world could this capsid binding lead to such a potent block. And so there's a lot of this drug is revolutionary and amazing, but I also really like the way that it ties into such basic science.
Yeah.
So the one of the things about what Brienne just described is that impresses me is that the capsid is relevant at all stages of infection, and that's highlighted in this figure that's in the paper. I know it's a paywalled.
Yeah, unfortunately,
so-
the paper, in keeping with another theme we're going to be picking up shortly, is paywalled.
Right. But it makes the point that because the capsid is relevant throughout the infection, an inhibitor that binds to capsid proteins can potentially inhibit at several places. This is semi off the record, guys, but I have a bone to pick with this figure because it's got the particle with a hexagonal structure inside and then this dotted line thing. Okay. And that, and then it's as if the dotted line thing is the capsid, but no, the hexagonal thing ought to be the capsid. It could just confuse the heck out of me.
Yes.
The capsid should be conical in
Mhm.
HIV.
And there's not two things, there's one thing. There
Yeah.
is a capsid.
Yeah.
There's
Yeah,
not a
the
green
red-
thing and a red thing inside it.
Yeah,
True.
that's right. The red thing is bogus. Yeah.
Yeah, okay, thank you. But
Yep.
the good part about that is that it made me go back and read up all about HIV structure
Yeah, for
and
sure.
et cetera to sort
I mean,
that out.
the good thing about the red thing is it shows something protecting the nucleic acid throughout the cytoplasm. That part's
Yes.
pretty true.
Right, and the nuclear import, I wanted to look up and make sure I understood that and that was true. And yeah, apparently the capsid makes it into the nucleus effectively intact, right?
There are some papers that have done some spectacular imaging on this.
Wow.
It's this whole set of experiments that have sort of all been done on this process are awesome.
That's a big chunk for the nucleus to swallow, seems to me.
Yeah. And so the way that it is interacting with the nuclear import proteins, they mention NUP, I think 153 here, is actually really key.
Okay. All right, cool.
So this has two indications. One for managing people with AIDS who are treatment refractory. They've had other drugs and they're not working anymore. That was approved in 2022 in Europe and also in the EU and also here in the FDA. And then for pre-exposure prophylaxis PrEP, the FDA approved that just this past June 2025. All right, so let's just describe how this drug came about and how it was
So
tested.
the PrEP, the way I see it, is in the current administration not an ideologically justifiable bit of health, right? So you shouldn't be exposed to HIV,
Right. Right, because you shouldn't be having sex.
right? Sorry.
I mean, this is not just there is heterosexual transmission of HIV
course.
in
Right.
the U.S. Okay, there's so yeah, it shouldn't be like Alan said. No more sex.
Right.
Well, I mean,
Right.
PrEP is for high risk populations. The point is,
in
you
theory,
shouldn't
yes.
be high risk,
Right.
right?
Well, but we
According
had a
to
letter
some. I'm not
yesterday.
saying
We
I
had a
believe
let
that.
we had a
No.
letter yesterday on clinical update from someone in the Washington Department of Health, okay, on the Pacific Coast, because I refuse to say Washington State, Washington Department of Health, and then people say D.C. said no, I said Washington, I didn't say Washington D.C. And she said they're seeing a huge uptick in heterosexual women acquiring HIV,
Hmm.
okay, and they've never seen this before.
Hmm.
So just revise what you're thinking. Of course, mother to child transmission as well, right? Anyway, so this drug was originally picked up in a screening campaign, and that identified a drug which inhibited the capsid, and then they optimized it further for enhanced antiviral activity, reduced cytotoxicity, and they end up with a drug with an antiviral potency between 20 and 160 picomolar. Pico. Man, usually nanomolar, we're happy with nanomolar, but this is amazing. It has a terminal half-life of 38 days, which is really long.
That's one of the really remarkable things.
Yeah.
AZT, the first HIV drug, had a half-life of 6 hours. Oh my gosh. So this is good.
The
So that's how the drug.
chemistry here is interesting. It's a remarkably complex drug, and I'm always interested in they identify a hit, right, and then they put the chemists to work on it, and they do all sorts of modifications and see if they can improve it. And they have a figure here that shows the starting drug and the ending drug, and it's just I can't imagine what the chemistry was like and what all the trials were like to get from the starting drug to this finished thing, because there's a lot of modifications.
So there turn out to be certain kinds of modifications that do specific things. And then the chemists know this, so they can apply that. But still, it's a lot of trial and error. You know what Dixon would call that final molecule?
What?
Rianne.
Dihydroxy chicken wire.
Very good.
Yes. Yes.
He would be saying that right now. Hey, that looks like dihydroxy chicken wire.
And it does. It's got, the panels of chicken wire on it.
Mm-hmm.
Got a lot of fluorines. 3, 6, 8 fluorine molecules. Wow. That must make it really stable, right?
I guess.
Because fluorine carbon bonds are hard to break down.
Can you imagine the process to make this?
Is it 6 or is it 10?
Sorry, what's that?
Is it 6 or is it 10?
3, 6. No, 10.
10.
There's 2
Yeah.
up there. I
Yeah.
missed. I missed the 2 on the
So many.
ring. Yeah, it's a lot. Also got a couple of sulfurs. Yeah, it's a pretty cool chemical. Anyway, so it's very effective in the IC50s and lymphocytes and in cells and culture. So how does it work? As we already said, it targets the core or the capsid, interferes with capsid assembly. You get malformed capsid structures. It inhibits nuclear translocation, and it also interferes with maturation. The particles bud off from the cell, and there is a post budding process that matures the particle, and this interferes with the conformation of the protein, so it doesn't mature properly. You get defective components. All right, so after oral administration, so there are going to be 2 ways of getting this. Oral administration, half-life can be extended to is 10 to 12 days, but if you give it subcutaneously, it's 8 to 12 weeks.
Yeah, it's really interesting pharmacodynamics here.
Very long. 8
Yeah.
to 12 weeks is huge. That's half-life.
Yeah, that's
Right.
half-life. So,
It's-
and there are a couple of dosage schedules they talk about in the review to use those 2 features. You've got the shorter half-life if you take it orally, and you've got the longer half-life if you give it subcutaneously. But a longer half-life often means that it takes longer to get up to therapeutic concentration. So I gather the regimens that are being proposed here or used are you start with a couple of oral doses, and then you also give subcutaneous dose. So getting onto it is multiple doses. But then once you've got the therapeutic level running, then it's twice a year, every 6 months. And there's like 2 weeks leeway in Like if you're late for your shot, it's fine. If you're within a couple of weeks, then you still maintain that.
And by the way, subcutaneous means you're shooting it into the fat layer
Yep.
that sits between the skin and the muscle below it. And now I fully confess I have the slightest idea what I'm talking about, but I wonder if that doesn't basically create a depot of this stuff in the fat that contributes to the long half life.
It may very well.
Because they do talk about, I mean, as we already said, right? The oral administration has a shorter half life.
Yep.
And that's going ultimately pretty quickly into the circulation. Okay.
The answer to your question is, so you look at the maximum plasma concentration, the Tmax. If you give it orally, it reaches Tmax in 4 hours. If you give it sub-Q, 77 to 84 hours. So it's taking longer to get into the blood,
Yeah.
and that must be because it's trapped in the fat. Right?
The other thing that to me is remarkable is that when you're on for the prep thing, the twice yearly doses are nearly a gram, over 900 milligrams. So that's what a gram is, and it's active in the picomolar range, which is 10 to the minus 12 grams. So I mean, it's phenomenal. You
Yeah,
load
it
people
is.
up with a gram of this stuff, and it lasts for 6 months because it's, I guess, released fairly slowly, metabolized fairly slowly, and works at a really low concentration. Remarkable.
Another nice feature of subcutaneous, well, oral administration, obviously you take a pill, but subcutaneous administration is straightforward. I mean, I don't know how straightforward it is when you're giving a gram of something, how many milliliters you have to
Yeah, I
actually
was
shoot
trying
in.
to
But
figure that out.
so that may complicate things a bit, but it's not the case where you need a trained phlebotomist to hit a vein.
Yeah.
You need to go into a fat layer, which everybody has somewhere.
I got
And so
plenty.
this is yes, so this is should be a straightforward administration that can
It's
potentially be done by people who don't have to be highly
it's
skilled.
1.5 mLs. Not bad.
Oh, that's not
That's not too
bad at
bad.
all. That might even be administrable by the patient.
So the cool thing, twice a year is easy because even a
Yeah.
pill a day, people have trouble with, right? It's at 365 days a year.
Yeah.
It's a problem, but twice, this is a big change. So there have been 2 kinds of clinical trials. So it was tested first in non-human primates and then went into 2 different kinds of clinical trials. One for the treatment of already infected people, right? So these are randomized clinical trials with people who never got treated and have HIV, or they are on treatment and they're virologically suppressed. And so in these trials, they um, define suppression as less than 50 copies per milliliter of HIV RNA in the blood. And there have been 4 phase 2 or 3 clinical trials. Some of them a bunch are still ongoing, actually. And when was this manuscript published? 2025. So yeah, those haven't been released yet. And these names of these trials are great: Calendula, Artistry, and the other ones are Purpose 1 and 2. So this first one, for example, they're giving either oral lenacapavir plus another antiviral, because you cannot take away the other antiviral for people who are being treated, and compared to without lenacapavir, right? And so the 96% of the patients with the combination had this suppression of viral RNA. And I believe that it's the same in the other treatment group, right? So it was 96% and 96%, 100% in the baseline antiviral therapy.
I think in a lot of these trials, the people on cocktails of one sort or another are failing treatment because they've
That's
turned
actually
up
another group.
multi-drug resistant.
Yeah, so that's a separate. This trial was in people who were doing okay on their therapy. And
Right.
They want
So
to say
they started off
throw in-
by taking people who were on therapy that was working, and they added this to make sure it didn't make anything worse.
Okay.
Right.
Basically.
And then the
And it
second
doesn't.
group, they have people who just tested positive but haven't gotten any therapy yet. Okay, and then they can give them this plus something else. And again, you don't get any bad effect of having lenacapavir in it.
So
Uh,
it doesn't inhibit any of the other
right.
drugs or interfere with them.
But then the Capella trial should have been a cappella, but
Yes.
it's the Capella trial in people with multi-drug resistant infections, right? And so they say one cohort, 36 people randomly received either oral lenacapavir or placebo plus their failing therapy. That's very interesting the way they're failing therapy. So they're still kept on their failing therapy, but they get either oral lenacapavir or placebo. And at day 15, the virological suppression rate was higher in the lenacapavir treated participants, 88% versus 17% in the placebo. So it really works, right? Where the other drugs are not working. Unfortunately, you do get resistance, as we'll see in a bit when you do this. One drug is always problematic, so you need to do multiple drugs. I mean, if you're failing treatment, that's usually because of resistance, and lenacapavir will help you for a while, but probably is not going to help you forever, unfortunately. And then they have PrEP trials, pre-exposure prophylaxis, and these I recently heard about. They're just amazing.
These are pretty jaw-dropping.
Yeah.
They have different we have 1, adolescent girls and young women in the Purpose 1 study, 2, cisgender men, transgender women and men, and gender non-binary individuals. I mean, none of this is going to be supported in the U.S. anymore.
Right.
And it's not going to be supported by USAID or other, agencies that we funded overseas because those have all been eliminated.
Because some people have ideological beliefs when all humans are equal, in fact.
Yeah.
It's ridiculous.
We
Right. So
should
that's
save everybody from infectious disease.
Yes.
you can. Then we have cisgender adult women, people who inject drugs, and then people who would benefit from PrEP. So you get twice yearly subcutaneous injection in these 5 studies, and there have been 5 purpose studies. 2 of them have been reported, and it's like one of them was 100% prevention of
It's
AIDS.
0 cases in the test arm. Yeah.
So we had 2,134 participants. The lenacapavir arm, 0 cases of HIV. And then the other one, 39 cases in the other antiviral combination that they use. So it's 100%. It's amazing.
Now, if I understood this correctly, some of these trials had a lenacapavir only arm, right?
I think so.
Yes,
And
that's correct.
it just occurred to me
It's
that
okay
in a
'cause it's
PrEP
PrEP,
trial,
right?
you don't have
Yep.
to worry about
Right.
resistance as much because you're preventing infection. You're not trying to treat an infection, so you don't have necessarily a lot of replication going on in the presence of the drug, which is where you're going to get
Yep.
the resistance.
But also, you don't have to worry about standard of care because they're not infected yet.
Right.
Yeah.
Well, that's also how you can test HIV vaccines, right? You have a population who's not infected. You give half of them the vaccine and half, and you tell them to go out into the world. And so then there was a Purpose 2 trial. So the one we just talked about, the Purpose 1, was in the adolescent girls and young women. The Purpose 2 trial is in cisgender men and gender diverse individuals from 7 different countries: US, Brazil, Thailand, South Africa, Peru, Argentina, and Mexico. 3,265 HIV negative participants. They're randomized to either get lenacapavir or a dual antiretroviral combination. So after 52 weeks, so a year later, 11 cases were found among this whole group, 2 in the lenacapavir and 9 in the other arm.
Yeah. So
So
that's
2
testing lenacapavir against conventional PrEP
right. So that's, there
instead
were
of
2.
testing it against nothing. So it's an
So you
important.
can see that depending on the population, you're going to have more or less cases. And also, lenacapavir is going to be 100% everywhere. In that first study, it was,
Yeah.
which is amazing. But even if it's 95%, it's really amazing too. So based on that, you have licensure as PrEP in many countries, and here in the U.S. Side effects, in case you're interested, the most frequent are nausea, diarrhea, and others, local reactions, not subcutaneous nodules, pain, erythema, etc., but no life-threatening things. Now, you do get resistance, and as you might imagine, the amino acid changes are in the capsid proteins, and they're near the drug binding pocket. Lenacapavir binds to a pocket in the protein, and these amino acids prevent or reduce its binding. And so they say you cannot use this as monotherapy. You can't just use it by itself. You have to use it in combination if you're going to try and treat patients
Right.
with 2 other drugs. But for PrEP, you can use it as monotherapy, and it works really well.
And it seems to me that the monotherapy aspect for PrEP is important because one of the advantages, of course, is that it's just a twice a year injection,
Yeah, it's great.
whereas the drugs that you would combine it with have to be taken more frequently, and so that advantage is doesn't go away completely, but it's diminished.
Now, the problem with this drug, so obviously, there are many countries in the world that need this, but can't afford it. This is $28,000 a year, as Gilead is the company that produces this. That's what they've priced it at. $14,000 a shot. Whoa. And so, UNAIDS is not very happy. They have written to Gilead and say, please drop the price. And they write, here's a quote from their article over at UNAIDS. UNAIDS has seen research that lenacapavir can be produced for just $40 per person per year, falling to $25 within a year of rollout. It is beyond comprehension how Gilead can justify a price of $28,218. If this game-changing medicine remains unaffordable, it will change nothing. I urge Gilead to do the right thing, drop the price, expand production, and ensure the world has a shot at ending AIDS.
And I provided a link here to the criticism section of the Gilead page on Wikipedia. They have a history of this sort of behavior of price gouging, especially with HIV drugs, and anti-competitive behavior and trying to delay introduction of new, better drugs that would compete with their existing drugs, and just generally putting profits ahead of people. So this is not a case where, well, they spent a lot of money to develop it and they have to recoup that. I mean, if you just look at the orders of magnitude difference between the production cost and what they're charging for it, this is not about recouping a reasonable profit margin. This is about grabbing as much money as you can before your patent expires so that you can pay yourself bigger bonuses.
Yep. Got expensive CEOs of these companies.
Yes. And Gilead is making money hand over fist. They're doing extremely well.
They make remdesivir.
They make a bunch of things. Yeah.
Rem was a big one. So hopefully they reduce the price. But when the Hep C triple therapy came out, it's the same thing, high prices. But
Yep.
they lowered it for certain countries that couldn't afford it, and hopefully
And
they
that
will do
was
the
that was
same.
Gilead as well.
It was.
Yes.
But
And
they did respond then, right?
well, kinda. So they lowered after all the public outcry, and they anticipate this. They
Yeah.
know there's going to be outcry. They lowered the price for certain countries, so you could get it for a lot less in India, like orders of magnitude less. But they simultaneously insisted on rigorous protocols that Indian doctors would have to use to make sure people were not shipping it back to the U.S., where they were
Mhm.
charging tens of thousands of dollars for it, and where health departments could not afford to medicate all the people they had who
Wow.
had Hep C, and so they're taking advantage of the way the U.S. healthcare system is broken in order to leech more money out of it. So it's not going to be cheap in the U.S.
But it's an amazing drug. It's a game changer.
It is an amazing
really
drug, and
cool.
the patent on it expires in 2041. So
Well, I hope that they uh, do the right thing, and I hope we figure out a way to get this to as many people as possible, because I think this could be very game changing if we can get it to as many people as need it.
yes,
The science that has spun off of the HIV pandemic is
Mhm.
remarkable.
Yeah, totally.
And still, by the way, we don't have a vaccine. Okay. People seem to think that, oh, vaccines. Yeah, we did that with COVID. You just, I can't use mRNA anymore. But still, you just make this thing and it's all okay. No, that's not the case. COVID, fortunately, was kind of low hanging fruit.
Yeah.
A lot of these things are a lot more difficult.
Every
And we
virus is
actually
different.
had like an infrastructure thanks to those HIV trials.
Yeah.
Yeah.
All right, our next paper it's actually suggested by Rebecca in Seattle. Hoping you will review this soon and help us laypersons understand it. It's incredibly dramatic, and the only background I have in understanding COVID and viral infections is by listening to you for the last 5 years. So, Rebecca, you ask nicely, but more than importantly, it's a really cool paper, really
Yes.
important paper. And, it's cancer. And I thought, we're not a cancer podcast, but we're scientists. We can figure it out.
And
And
it
it's
is a
got
virus
viruses.
paper.
Yeah, there are viruses in it. There's a lot of immunology as well.
I learned some cool cancer stuff here that I didn't understand before. I didn't
Yeah.
appreciate.
Yeah, me too. Yes, I did. Totally learn new stuff. So this is a Nature article, Respiratory Viral Infections Awaken Metastatic Breast Cancer Cells in Lungs. A long list of authors.
Yeah, two
Oh,
co-first
look, it's on the
authors.
front page, Alan. Look.
Yeah. They finally fixed it for me. Thank you. So two first authors, Shi Chia and Brian Johnson, and 4 senior authors, Rolf Vermeulen, Julio Aguirre Guiso, Mercedes Rincon, and James de Gregory. And they are from University of Colorado, Utrecht University, Imperial College London, University of Connecticut, Albert Einstein College of Medicine, COVID-19 International Research Team in Medford, Mass, University of Pittsburgh, Broad Institute at MIT and Harvard, and Johns Hopkins, the Van Andel Research Institute in Grand Rapids, Michigan, Children's Hospital, Philadelphia, and UPenn. Big team.
So this is mainly about breast cancer, which is the most diagnosed cancer in women, the second most common cause of cancer-associated deaths in the U.S.
Number
And
1 is
so-
lung cancer, by the way.
Number one is lung cancer. Second most cause, yeah, behind lung cancer. And so, if you enter remission after breast cancer, it turns out that there are dormant disseminated cancer cells, which we're going to call DCCs.
This was a new
All right.
concept to me.
Yes.
Me too. I didn't know this.
Yeah,
But of
I
course,
thought
if we were,
metastasis was something that happened, and like that was part of a continuous process. And we didn't
Yeah.
get the cancer early enough and it metastasized. But somebody in remission, you think, it's all gone. Well, no. In remission means it's all stopped and you can't detect it. And apparently it's pretty common for breast cancer survivors to have these dormant
Yeah.
cells just hanging out in other tissues like the
Yeah.
lung.
At the same time,
Lung,
I'll bet
bone,
you
and liver.
everybody has some knowledge of a story of somebody having recovered apparently from breast cancer, and years later, all of a sudden,
Yeah.
they have a relapse.
Yep.
And apparently, dormant is a key word here,
Yes.
right? And disseminated. These cells are disseminated, and they're not doing anything. They're just sitting there waiting for an opportunity.
Which is key because detection is often based on their ability to do some metabolism, particularly some quick metabolism. And so,
They also
because they're dormant, they're not going to be detected.
have to
Right.
be. They also have to be a certain size, even on a PET scan, right? Which is metabolism. They have to be
Big
a
enough to see.
centimeter or something.
Yeah.
The
Centimeter.
dormancy itself is interesting. They don't talk
Very
too much about that, but how do you do that? Just hang out for years.
Right. They're
Yeah,
like it's like a neuron, it's just sitting there.
They're not dividing. Yeah,
Yeah.
they're just
Right.
sitting there. Which this happens in bacteria. They remain dormant. It's called non-viable but culturable, or non-culturable but viable. I forgot which way, but they're not dividing, so you cannot culture them and say you have this infection, but they're there, and under certain conditions they'll start dividing again. So anyway, these breast cancer dormant cells can go to the lung, bone marrow, and liver, and they sit there. But as you said, Alan, I always thought that you have an initial tumor and you get some metastases, and then you hope you take care of it with surgery and chemo. But I always assume you just don't chemo everything, and there are a few hanging around, and then within a year, but no, this is a long-term thing, and they're
Yeah,
dormant. This is really different. I don't know how general this is among cancers, right? Because this is just breast cancer
right.
here.
And it's important to point out that everybody is generating cancerous cells all the time throughout your
Mhm.
life. And normally your immune system comes along and says, and deletes them. But these dormant cells presumably get around that by just not doing anything.
Yeah, that was one question I had that I didn't know enough about, was whether these dormant cells are avoiding immune detection because they're, not actively producing factors to be recognized, or if they're actually immunosuppressing.
Mhm.
And there are lots of tumors that are known to be immunosuppressive, and so that was sort of one of my first questions, was how's the immune system letting these slip through the cracks?
Yeah, we're gonna find out a little bit about that in this paper. So the question is, what makes these cells, these dormant cells, come back and not be dormant anymore? And the perturbations like inflammation is one of them, can cause these things to re activate and then cause nodules and so forth. So what causes inflammation? A lot of things cause inflammation, but respiratory
Yeah.
infections do it. Viral respiratory infections, right, which are very common, influenza, SARS-CoV-2, and if these get into your lung, you get inflammation. And so if you've got these dormant cells there, the question is, can viral infections make the cells come out of dormancy?
And then in 2020, the world did a natural experiment that
That's right.
it turned out death rates from cancer went up,
Yep.
significantly in the databases. The first couple of years of the pandemic, before the vaccines were in widespread use, and once you correct for everything, like there was a pandemic going on, people weren't getting their regular screenings. You still have extra deaths from cancer. Like,
Right.
why are people suddenly dying of cancer when we got a respiratory pandemic? And that the theory here is maybe it triggered reactivation of metastatic cells.
So in this paper, we're going to look at it in two ways. We're going to look in mouse models. They have a very nice mouse model for this dormant cells after breast cancer induction, and then we're going to look at data from the COVID pandemic. Okay, and so first, and they're going to look at influenza virus and SARS-CoV-2 in this mouse model. So they have mice that have breast cancer, and they are a model for metastatic dormancy. We know that. So it's a mouse that overproduces HER2, which is epidermal growth factor receptor 2, which is a marker for breast cancer, and it's often the gene is mutated, and so keeps signaling, and that's how it's the cancer driver. And you can do that in mice. They will develop breast cancer, and then it will form these dormant cells in the lung. So it's a beautiful model. They're very lucky it has this very nice model. So basically, you get early lesions in the mammary glands, and these seed the lungs and other organs with these DCCs within 10 to 14 weeks, and they are dormant as single cells for up to a year, and then they progress to metastatic disease. And so other people have developed this and studied it extensively. So it's a perfect model, right? Because mice only live 2 years or so. And so this is a model for dormant DCCs in the lungs and bone marrow of people who are in remission for years to decades. So what they do is they take these mice and they infect them with influenza virus, sublethal dose, so they don't die, right? And they both, the control is normal mice, wild type mice, and these um, HER2 producing mice. Both get an inflammatory response. You get cells going into the lungs, right? Viral clearance is pretty much the same, so nothing overtly different between these two mice. And then they look at different times after infection 3, 6, 9, 15, 28, and 60 days after infection for cancer cells, HER2 positive, because that's the oncogene, right, that's in these mice, in the lungs. And people have already shown that this happens. You get isolated DCCs in the lungs, but when you infect with influenza virus, this metastatic burden increases between 100 and 1,000 fold. And that number of elevated number of pulmonary tumor cells remains high at 28, 60 days and 9 months after infection. So there's a clear effect of influenza virus infection. And they also do it in a different mouse background and get the same results. There's no effect of influenza virus infection in the mammary gland. Because one argument would be, well, the infection is increasing the numbers of HER2 positive cells in the mammary gland, and that's what's going into the lung. But that's not it. The mammary gland stays the same. It's the lung that is seeing this effect. So that's one model of a mammary tumor in mice. There are two other mouse models that they have. One is a model where the polyoma middle T is the oncoprotein that's driving the tumor, right? So it's the HER2 protein, oncoprotein in the first model. Now it's the polyoma middle T, and this is going to make Rich very happy because polyoma comes back again, right? And these also get breast cancers, and the reason is they're using a mouse mammary tumor virus promoter to drive this, so it's mammary gland specific. And they find the same things with the polyoma-driven tumors, and they also use another model to have cells, orthotopically implanted cells. These are cells, breast cancer cells that they can implant into the mammary gland, and you get the same effect. They become DCCs in the lungs, and it increases with influenza virus. So in 3 different models, influenza virus increases the numbers of DC. They reactivate the DCCs, and they begin to multiply and metastasize.
Yeah.
It's very cool.
All very cool. One, this is not a, I wish, this should be in the paper kind of thing, but one experiment that would be really interested in, sort of beyond this, is that they did an intranasal influenza challenge, and I'd love to see a virus outside of the lung, because I think the idea here is that these dormant cells have moved to the lung because that's one of the places they like to move. And so what if you had infection and all of these immune responses happening some other location, not in the lung? How much does that influence the proliferation of these cells? I think the idea that I would get from this is that the idea is that it's a lung-specific virus is causing this lung-specific reactivation. But if they looked at a virus that was tropic for some other anatomic location, that could be cool.
Yes. Right. To know if this is some general effect, you had this effect on the immune system systemically that caused these cells that happen to be in the lung to reactivate, or was it a lung-specific reaction? Yeah. Good question.
That gets at that same question about the cells in the mammary. Are you getting proliferation in the mammary tissue, or are you getting proliferation in the lung? I think that looking at a infection that was somewhere else could be a neat thing to try next.
Give them a GI virus or something.
There's in terms of tissue specificity, there are hepatotropic mouse viruses,
Sure.
right? And there are DCCs in the liver, right? So that would be interesting.
So next they try and tease out what's going on, how infection is doing this. And the first thing they say, okay, are these DCCs actually triggered by infection? And what they use is a marker in the cells called Ki67, which is essentially a marker for every part of the cell cycle except the dormant phase, which is called the G0, right? And so they can stain for Ki67. They can also use incorporation of the thymidine analog to measure the awakening of the cells. And by both measures, they show that after influenza virus infection, the cells start to divide, right? Thymidine is incorporated into dividing DNA, so that's one way to look at it. The next is that, what do these cells look like, okay? These DCCs. So the dormant ones in these mouse models, they look like they're called in a mesenchymal state. All right. And it's driven by a protein called ZFP281. If ZFP281 is lost, that makes the cells shift to an epithelial state. All right. So mesenchymal state is characterized by the presence of vimentin, a cytostructural protein, and then the epithelial state is characterized by cadherin. Right. So the shift from the exit to dormancy goes from mesenchymal to epithelial like. And so they look at these markers and they show that in fact during influenza virus infection there's a shift to losing mesenchymal markers, and then there's an epithelial shift. But what they find with influenza virus is that there is actually an unprecedented mixed population of cells, both mesenchymal and epithelial cells, which is usually not the case in without the virus there. They call it a unique and, to our knowledge, previously unrecognized hybrid and proliferative pattern with increased expression of both mesenchymal markers and epithelial markers. Okay. They also find, so they look at a lot of antigens by a variety of techniques that are produced in these cells as they're undergoing this shift after infection. You find activation of um, proteins involved in angiogenesis, right? Production of blood vessels, which you need to feed a tumor, a lot of collagens, um, and other genes implicated in angiogenesis. But they also find a decrease in a protein required for antigen presentation by MHC. So many pages, MHC1. So they say maybe this is part of an immunization thing, like Brienne, you were saying.
Yeah, so it's both
earlier.
that decrease in beta 2M, which is part of antigen class 1 presentation, and also an increase in CD274, which is PDL1, which is also potentially part of that immune evasion.
So the PDL1 would cause the T cells to get exhausted, right?
So basically, if the PDL1 on the tumor cell means that if a T cell interacts with that tumor cell, the tumor cell would be like, you have to be turned off. And so the tumor cell would actively turn off any T cells that interacted with it.
Right. Okay, so then they look at IL-6, a pro-inflammatory cytokine often induced during viral infection. They're also known to promote cancer, malignancy, and metastasis, right? And this is something I didn't know either here. IL-6 produced during acute inflammation resulting from biopsy or chemotherapy contributes to the development of lung metastatic outgrowth of these mammary tumor cells. Can you imagine? You get a biopsy and that contributes
to the problem.
You get chemo and that contributes to the. How does anyone ever survive? Holy cow.
Yeah.
And that's.
Yeah. A lot of these inflammatory cytokines do have this ability because, and sort of the idea is that they are producing, they're inducing proliferation. Because if you think about an area of inflammation, one of the things that needs to happen is you need to get some wound healing. And so the tissue needs to, you need to have some proliferation and you need to get some new cells in that area to heal the wound. And so there does seem to be a proliferative effect of some of these cytokines that has been noted in a number of tumor models.
So they look at IL-6 after influenza virus infection in these DCCs. It clearly goes up. And the source is epithelial cells, which are the ones infected by the virus. So is this IL-6 needed for the awakening? Was there a movie called The Awakening?
Or Awakenings, maybe?
I think so.
Yeah.
Maybe a book.
The Awakenings. Anyway, is IL-6 involved? So they use IL-6 knockout mice. They don't produce IL-6, right? You still get this reawakening. So you don't need IL-6. It isn't required for the dissemination to the lungs for sure. But if you're looking, I'm sorry, so I jumped ahead. You don't need IL-6 for the establishment of the mammary tumor or for the dissemination to the lung. But when you do the influenza virus infection and look at the reawakening, you need IL-6 for that. It's dampened in the absence of IL-6. Okay, but um, it gets more complicated, right? So they take mammary glands out of these mice. They grow them in culture. They treat them with IL-6, and you can see that they proliferate. These cancer cells proliferate, and so they're affecting the tumor cells directly. IL-6 is affecting the tumor cells directly, and so they think this plays a role in the reawakening. So IL-6 plays a role in the reawakening, or the awakening, I should say, of dormant DCCs. But it's going to get more complicated.
I know. And this is where it's like, what? This is where I couldn't believe it.
But even though IL-6 seems to be essential, you don't find much of IL-6 in the bronchoalveolar lavage of these mice 15 days after infection. And so they're thinking maybe something else is here, which is good science, right? You shouldn't just stop at IL-6. It could always be something else.
Don't just go with the first answer that seems to make sense. Yeah.
Right. So they look at what else is going on here in the lung when you infect with influenza virus. So of course you get neutrophils coming into the lung, you get CD4 T cells, you get CD8 T cells, and you get B cells. All these cells are coming in the lung to try and deal with the infection. And so they focus on the CD4s. They say, do you need CD4s for this awakening? So they do CD4 cell depletion before infection, and then they look for the metastases in the lung section. So if you deplete CD4s, you reduce the number of awakened. Like, it's totally counterintuitive, right?
Yeah.
What is going on? CD4s are supposed to be good. So you reduce the number of awakened DCCs by 28 days after infection, but not at 9 days. Okay. So they're thinking IL-6 is important early, and CD4 are important late. And if you deplete at 10 days, you also decrease the number of DCCs at 28 days. So they say CD4 cells contribute to the maintenance of awakened DCC. So the IL-6 initiates the awakening, and then the CD4s take over and do it later on in infection. They looked at neutrophils and neutrophil extracellular traps, which we've talked about on immune. These are when neutrophils throw out traps of DNA, right, to grab viruses and bacteria. They don't seem to make any difference here after influenza virus infection. And also, depletion of CD8s had no effect in the presence of these DCCs. Okay, so they didn't give up on CD8s. They found that when even though you get depletion of CD8 doesn't make a difference, if you deplete CD4s, you get more CD8s accumulating in the virus-infected mice. This is a little complicated. I hope I'm not losing people, right? So CD8's depletion didn't make any difference, but if you deplete CD4s, you now have more CD8s.
And okay, so just to recap a little bit here, IL-6 is the alarm clock. CD4 appears to be the snooze or the cup of coffee, maybe. It's going to keep them awake.
Keep them awake, yeah.
CD8 and CD4 and CD8 cells, CD4 we think of as the helper cells, CD8 as the killer cells. So it's two different types of response you could get. And here we're seeing that CD4 is going to make things worse in terms of making the tumor grow. CD8 may actually be something a tumor doesn't want.
Yeah.
Right. And it may, again, the idea that the tumor could get killed by CD8 cells is something that is consistent with a lot of tumor immunology. As I look at this, I kind of feel like it's saying that maybe somehow the CD4s are keeping the CD8s out, or not letting the CD8s in.
Yeah.
Though that's sort of not super clear.
Yeah, they say maybe the CD4s repress recruitment of CD8s.
Does that make any sense to you, Brienne? Is
No.
that so? CD4s managing CD8s is not Not in the
I
textbooks?
I know about ways that CD4s can positively manage CD8s, but this sort of more negative thing I am not familiar with.
Okay.
So then they decide, let's deplete both CD4 and CD8. And this partially restores the numbers of HER2 cells in the lung. So the numbers of HER2 cells are their measure of reawakening, right? So um, you need CD8s. That's the key, and CD4 is something somehow interfering with it. So then they ask whether it's the DCCs that are reprogramming the T cells to behave in this weird way. And for this, they use the HER2 mice again. They infect with influenza virus, and they look at a variety of responses. But one of the genes that they find that is highly upregulated after influenza virus infection in the CD4 cells relative to wild type mice is a protein called tumor necrosis uh, factor alpha IP3, which encodes a ubiquitin ligase. And this normally over suppresses the anti-tumor activity of T cells, CD8 T cells. And then there's another protein which was downregulated that is also upregulated, which restrains CD8 cell activation and expansion. So I have the induction of basically two proteins that are dampening CD8s by CD4 cells, right? So they're made in CD4 cells. So that could explain the suppression of CD8 cell activity by the CD4s.
So it's sort of like the DCC is telling the CD4 cells to inhibit the CD8s.
Yeah, how that does it is the question, right? We
Yeah.
don't know how that happens. That's the key, but looks like CD4s are influencing the CD8s. Yeah. So they say these results indicate an impaired effector cell phenotype in CD4s from these mice relative to wild type mice. But how that happens, I don't know. But they say it looks like a bias to a more memory phenotype and increased survival with less effector function. So somehow maybe the DCCs are in pushing the CD4s towards memory.
Yeah, so somehow the CD4s are pushing the DCCs towards memory, or I would just say towards a phenotype that's going to inhibit or exclude CD8s.
Yeah.
So the DCCs are sort of, telling the CD4 cells to get rid of those nasty CD8s.
Other genes involved here are so there's some genes that were reduced in CD4 cells that are needed for T cell activation. Also, they observed that as well. So again, adding to that story. So they do what's called pathway analysis. So they do RNA transcriptomics, single cell RNA seq. So you sequence RNA from single cells that you isolate from the lungs, and then you ask what are these genes involved in, and so the computer tells you what pathways all these genes are involved in, and they find that the presence of the DCC skews the macrophage phenotype in the lung and also impairs CD4 and CD8 activation after infection, and that favors the persistence of tumor cells. So they say the effect of CD4 cell depletion on eliminating DCCs seems to be mediated by enhanced CD8 responses against them. So the CD4s are inhibiting the CD8s, which would otherwise clear the tumor. And very interesting.
Yeah.
So they did an experiment to look at whether these CD4s are suppressing the activation of CD8 cells. So they isolate CD8 cells from the infected mice or from CD4 cell depleted mice at different times. And they look at the ability of these CD8s to kill mammary tumor cells ex vivo using cultured of cells. And the CD8s from the lungs of mice where they depleted CD4s had great tumor killing activity. but not when you isolate them from cells, from mice that maintain CD4s. So the CD4s do something to the CD8s such that even if you pull them out of the mouse and throw them into culture, they can't kill tumor cells very well.
So it's interesting because now that I think about it, I can certainly think of a way that CD4s could inhibit CD8s. I just don't think of it that broadly because there are different subtypes of CD4s, and one of them certainly could influence CD8 T cells. But there are a whole bunch of really famous genes that are involved in that situation, and none of them are mentioned here. And so now, as I'm looking at this, I'm like, oh, I want to go through their raw data and actually look at what's going on with the genes that that suddenly occurred to me. I think it didn't even occur to me that was a option here because they so much don't mention all of the usual suspects in that situation.
Do these effects of the CD4 cells require contact with the CD8s?
They can.
They don't have to, though.
Yeah.
Okay.
That's a hotly debated topic.
All right, so then they look at SARS-CoV-2. What do you think the result's going to be?
I think it's going to be the same.
It is the same. They do same experiments in the same mice with SARS-CoV-2, a mouse-adapted SARS-CoV-2, and essentially find the same thing. These can reawaken the metastatic cells in the lungs of mice, and it requires IL-6, as we saw before, to get it going. So that's all the mouse experiments. Then they say, what about people? Do we have enough data? And so, as Alan said in the beginning, we had the pandemic, right? They say it was a unique opportunity to study the effects of infection on cancer progression, because during the first years of the pandemic, we were collecting data on lots of people, right?
And
so
you knew
there's
what virus they'd been infected with and when. That's the
Now-
critical piece that was missing from every prior,
Yes,
flu year.
Yes, that's right.
So
there's
you
a-
say
there's a
we
data-
were collecting data.
It's actually the UK.
Exactly, because they have a public health system where they can keep good
Yeah.
records and they can mine that for data.
And
Wouldn't
because
that
they
be
were
nice?
they were good at testing early on.
Right.
Yeah.
It's called the UK Biobank. And so you can ask if you had a SARS-CoV-2 positive test result among a population of cancer survivors, right? So you've already been diagnosed with cancer beforehand. Getting infected have an effect on you. Did it increase your risk? It actually looked at all-cause non-COVID deaths and cancer mortality. Okay, so they looked at people with PCR tests before December 2020. Because that's when vaccination started, right? Although I would have loved to know the effect of vaccination on this,
Sure.
right? Would have been really cool. But they just looked at before 2020, that first year when there was no vaccine. So they have, so then they follow up these patients for 2 years.
And these are people who
31st.
had a cancer diagnosis before 2015. So
Right.
they had cancer. You know now that they're in remission. And so they
Right.
should be the model that you're looking
Right.
at.
So you got 4,837 participants in this group, and they had 413 deaths, 298 deaths in the test negatives, and 115 in the test positives. So that's an odds ratio of 4.5. And they excluded COVID-related deaths, and they still see that SARS-CoV-2 positive cases had increased mortality, odds ratio of 2.56. So a twofold increase in cancer mortality in people who tested positive compared to people who tested negative. Odd ratio of 1.85.
I don't know that you could do this, but it would be sort of interesting to know how much of that were, say, breast cancers versus other types of cancers. And again, I don't know enough about these DCCs to know if they're involved with all cancer types or where they go. But in theory, this is some kind of DCC in the lung getting reactivated by a respiratory virus. Is this relevant for all cancer types? Or if you somehow could just look at the breast cancer patients, would you suddenly get an even bigger odds ratio?
Need a bigger
Mhm.
database.
So they then looked at people who had a cancer diagnosis before January 2010, just to see how that analysis would do compared with theirs. And they have fewer cases, so there's loss of power. But you see the SARS-CoV-2 positive people had increased risks for all-cause mortality, for non-COVID mortality, and for cancer mortality. Now, non-COVID, Daniel always talks about this. You know, you get SARS-CoV-2. He says you go in the hospital and you leave, and 30% of those people die within a few months of, you know, heart problems or other problems as well. That's the all cause mortality. But that's still an increase in the cancer mortality there. And then they reduce the follow-up period to December 2020. The odds ratio increases a bit. So they say um, the strength of the association between SARS-CoV-2 infection and deaths from cancer is the strongest shortly after infection and goes down with time, right? So that's good news, because if you had a SARS-CoV-2 infection and a previous cancer diagnosis and
If
you're
you're still
still
around,
alive,
you're probably out of the woods from that.
you're out of the woods with
Yeah.
that. I mean, remember, this is not 100%. This is not like 100% of people who have a cancer diagnosis and get
This is
SARS-CoV-2 or
something that
HER2.
can happen. It's
Can
not
happen.
Right.
something that
And in
will
fact,
happen.
the numbers are noticeable. In some cases, not even statistically significant. There are trends, but
Yeah.
They're trends.
but it's not huge. I'm thinking about Rebecca, who wrote in. Okay,
Mhm.
you know, it's a risk factor,
Yes.
but it's not a death sentence.
I
Right.
mean,
Mhm.
I would do all you can to avoid getting a respiratory
Yeah,
virus infection.
Yeah.
Sure.
might even
If
get
you're in
an mRNA
remission.
vaccination.
Yeah.
Mm-hmm.
What's that? What that?
I said a dirty word, mRNA.
So I would get vaccinated. I would wear masks in crowded situations, even in the summer, right? I'd just be careful. I don't, because it's an increased risk, right? Anyway, they do also have another database, the Flatiron Health Database, 36,845 female patients with breast cancer. So this is now narrowing it down to breast cancer. Um, and so, again, a lot of information in this database. Did they have, after their primary diagnosis, did they have an increased risk of progression to metastatic disease after COVID-19? Um, hazard ratio of 1.44. So a little lower than we saw for the overall cancer, but still an increased ratio of risk of dying after SARS-CoV-2 infection.
And the
And what
Flatiron database, by the way, I mean, to Rich's point, we do have databases in the US. And this is one of them. It is a private company. I gather they're drawing from insurance company and electronic medical record data, and this is specific to cancer patients.
they point out that this database does not contain test negative data.
Right.
So um, there may be undiagnosed cases. Maybe it's underestimated, the hazard ratio. That's the whole. So basically, in patients with breast cancer, COVID-19 increases lung metastasis risk as well, just like in the mouse model. So this is a beautiful illustration of how you can use a mouse model to understand mechanisms, and then you can do observational studies in people to see if it applies, because it need not apply to people, right?
Right.
What you find, because mice lie.
Apparently, these mice are telling the truth.
These mice are on the level.
Yeah.
And one of the things that interested me here about these, I mean, besides, oh my gosh, these cells exist, these
Right,
these
right.
metastatic cells just hanging out. They're dormant, but then when they reawaken, they remember everything from their past life as a tumor. They immediately go to, the angiogenesis and the immune suppression and
Yeah.
the whole program just
Yep.
boots
Yep.
right back up again with the trigger, which apparently IL-6 is involved in.
Yeah.
Yep.
Yeah. I mean, as I look at this, I come up with all sorts of questions that I am interested in. Does, having infection after previous vaccination, as
Yep.
is
Yep.
happening with many people now, protect you? What one experiment that I think I would really love to see done is to look at vaccination just in general, to show whether or not vaccination increases these cells reawakening, because again, you are turning on an immune response. This is one of the reasons why I think that the location is so important, because when I get an mRNA vaccine, I don't get it intranasally. I'm not getting necessarily a set of lung inflammation, and so potentially in that case, I might not reawaken cells in the lung, even if I might have if I got the lung infection. You could also say, well, maybe it's a different, it's more controlled, it's more inflammation, more brief inflammation, maybe wouldn't lead to the same IL-6. And so I think that showing whether or not location matters and whether or not vaccination matters is going to be important for people to have for that data. Whether this is sort of a breast cancer only thing, I don't know enough about DCCs to know anything about that. Whether vaccination is protective against this if you do get that respiratory infection later. I think there are all sorts of really cool experiments that I hope people are doing after looking at this.
I mean, it is possible that intramuscular immunization gives you IL-6 that gets in the circulation and goes into the lungs, right?
Maybe.
Needs to be looked at.
It needs to be looked at.
Yeah. Good point. I'm not talking about mRNAs, by the way. I'm just talking about any vaccination would induce inflammation and could trigger reawakening. Who knows?
It could, but at the same time, I'm sure that cancer patients are experiencing many infections with many different microbes following their cancer diagnosis.
Yeah, so that's gonna
and so it doesn't seem like every infection can do this, because if that was the case, we wouldn't have remission.
Right.
Yeah.
By the way,
Yeah.
Awakenings is both a book and a movie. The movie based on the book, and the book is Oliver Sacks's experiments with the new drug L-Dopa on a population of individuals in a hospital in the Bronx that were experiencing a Parkinson's-like disease called encephalitis lethargica. That was a mysterious epidemic in the 1920s that basically put a bunch of people to sleep in a Parkinson's-like thing. And the L-Dopa woke them up and they went, whoa, who am I? Where am I? What's going on?
I think it was just for a while.
Yeah, just for a while.
And I think Robin Williams was
Robin Williams was in the movie, and Robert De Niro.
Wow.
I haven't seen the movie. I've read the book.
So this is, let me summarize this. So we have these respiratory virus infections. They promote expansion of dormant cancer cells, in this case in the lung. And there's two stages. There's an IL-6 dependent switch from a mesenchymal phenotype to a hybrid state, which promotes the expansion. Then we have a return to quiescence and an establishment of CD4 niches that inhibit elimination of the DCCs, partly through suppression of the CD8 cells. So the question is, what could you do? They talk a little bit about developing interventions to minimize this. So you have millions of cancer survivors, right, who get respiratory infections. So this can be a significant morbidity and mortality, right? So, we have anti-IL-6 receptor antibodies that have been approved by the FDA, also JAK1/2 inhibitors. Maybe they could be trialed to see if they mitigate any of these reawakenings or developing other therapies as well.
Yeah.
So thank you, Rebecca, for pointing this out, because I learned a lot.
Great paper.
It's very interesting.
And this paper is open access, by the way.
Yes.
That's cool. I was wondering, so we talked about vaccination. Do you need to have disease, or is infection enough to do this? I would guess you need lung disease, because you need those.
You need the immune response to be angry enough, right?
Yeah, that's sort of all part of what I'm trying to figure out here is, how much response and what kind of response is.
Yeah, I think
really
the questions that you pointed out, Brianne, that interest me the most are the how localized an effect this is, and uh, in the same vein, what's what's the role, if any, of vaccination? Because that's, in terms of localization, that's a different kind of phenomenon.
And you
So
might even see, if you're vaccinated and you get the infection, there might even be kind of a sliding scale of risk depending on how immune you are. I mean, maybe your flu shot totally protects you and you don't even sniffle. Maybe your flu shot you get a little sick, and that might you'd need a big database to look at this, but these big databases are coming
Yeah.
online.
It looks like Rebecca also had a question about whether, taking an antiviral.
I wrote that in my
Oh,
stuff.
sorry.
Yeah, that's a good question. Is an
But yeah,
antiviral
what's up with would an antiviral reduce disease enough to
Yeah.
reduce this? Looks like Vincent has a great question,
Yeah.
Rebecca.
Yeah, I just jotted those down so I wouldn't forget them. All right, let's do one round of emails or so. I want to take this first one.
I have something at 5, so
Okay.
somebody's going to knock on my door.
Yeah, one. I think we can do one
yeah,
round.
Yeah.
Um, this is from Volker. I just listened to episode 1235 and was surprised that sanitation did not feature in the discussion about polio eradication. As I understand it, countries using only IPV, which does not induce mucosal immunity, still report almost no polio in wastewater whenever they do analyze it, despite recurring importations from regions where polio is circulating. To me, this suggests that the effective reproduction number remains below 1 in these settings, likely due to sanitation and hygiene standards interrupting fecal oral transmission. Otherwise, we should expect ongoing silent transmission and detectable polio in wastewater given the absence of mucosal immunity. From this perspective, it seems that eradication in low income countries cannot rely on vaccination alone, but also requires improvement in sanitation to drive R below 1, whether in combination with IPV, OPV, or transition strategies. If you see an alternative explanation for the scarcity of poliovirus in wastewater in IPV only countries that does not rely on sanitation, I would be interested to hear it. Best wishes and greetings from Bavaria. So I sent this to a person who shall not be named. Um, and here's the response. First, it would be greatly appreciated if one does not refer to virus reproductive numbers, as they are not an intrinsic property of any virus and its meaning is dubious as best. The continued transmission of polio in low income countries is due to poor vaccination coverage and some misunderstandings about poliovirus transmission and immunity. Sanitation conditions in these countries have significantly improved and poliovirus continues to circulate. Outbreaks of paralytic polio in the U.S. during the 1900s were not due to poor sanitation conditions, but a consequence of poor population immunity and the fact that you can productively get reinfected by the same serotype, i.e., the virus reproduces and is shed in those individuals. This fact remains true for all vaccinated individuals. The reason for the outbreaks of the 1900s was that improved sanitation conditions delay when children are initially infected by the virus. In fact, when sanitation conditions remained poor, there were fewer cases of paralytic polio. Children living with improved sanitation conditions were generally infected when they were no longer protected by the presence of maternal antibodies. As one ages, the probability of developing paralytic polio increases. In many of these low income areas where polio continues to circulate, it's likely to be found circulating within vaccinated populations. The role of mucosal immunity for reducing virus shedding is unclear. Early work examining virus shedding in infants following OPV administration suggested that gut immunity reduced the duration of virus shedding. The studies done in older populations suggest that mucosal immunity does not affect virus shedding. IPV or OPV vaccinated adults shed virus with the same kinetics. Furthermore, there are data that suggest IPV can induce mucosal immunity or that IPV stimulates antipolio immune cell migration into the nasal pharynx. The detectable level of mucosal immunity in these cases is thought to depend upon a number of doses of IPV one receives. However, because mucosal immunity is underneath the level of detection by our lab assays, does not mean it is not present and functional. Nevertheless, in their recent CID editorial, Plotkin and Chumikov correctly state that mucosal immunity fades. Studies in the 60s, when virus circulated, revealed that IPV reduces the duration and magnitude of virus shedding. However, now that virus circulates in only a few places, the probability of someone shedding following IPV immunization is similar to that of unvaccinated. If you do shed, the kinetics are similar to OPV. The scarcity of polio detection in wastewater in IPV-only countries does not rely on sanitation, i.e., sewer systems, but high vaccination coverage and high population immunity. If you were to stop vaccinating in IPV countries, you would see outbreaks of paralytic disease and extended virus transmission and circulation. Sanitation in these countries would not have changed. This week alone, 8 environmental samples in Germany were positive for circulating vaccine-derived polio 2, and 6 sites in Israel were positive for circulating vaccine-derived polio 1. The last positive tests reported were in early 2025 for Germany and December 2024, when a patient in Israel was diagnosed with paralytic polio due to circulating type 2 infection, vaccine-derived type 2 infection. Did all the toilets and closed sewer systems just overflow this week? Little humor there.
Yes.
IPV-only countries infrequently survey for poliovirus, as well as on a country level, have high vaccine coverage and population immunity. Poliovirus is imported, and more often than not, the virus burns itself out by acting as a booster by infecting vaccinated individuals. Consequently, it circulates for a very short period of time. Only when the virus circulates in an under-immunized population and a case of paralytic disease is detected do we know of its circulation. Very good.
So, we often say that IPV does not induce mucosal immunity. I'm hearing something different here. Is that right? Am I hearing this correctly?
It can,
It
yes.
can.
I mean, especially in a previously OPV immunized cohort, when you give them IPV, it boosts mucosal immunity. It's very interesting. Yes. All right. Who's a Frank Zappa fan?
You can count me in.
All right. Read the next one, Rich.
David writes, were Zappa still with us, he would be posting this online. Some scientists claim that hydrogen, because it is so plentiful, is the basic building block of the universe. I dispute that. I say there is more stupidity than hydrogen, and that is the basic building block of the universe. There is more stupidity than hydrogen in the universe, and it has a longer shelf life. Frank Zappa. And the author here, David, says, me, it's like the law of the partial pressure for gas. Get rid of science to make more room for stupidity. And he's got a link here to a site that has 25 of the best quotes from Frank Zappa. And I highly encourage anybody to read through this because it's just awesome.
Yeah.
The universe consists of 5% protons, 5% neutrons, 5% electrons, and 85% morons.
Beautiful.
Yes.
Mine is like a parachute. If it doesn't work, it doesn't work if it's not open.
Yep.
Wow.
Very good
This is
stuff.
good.
Frank Zappa was a genius. Have you seen this old? There's an old video clip of Frank Zappa on, God, what was the? Who was? I forget one of the original late night shows. At any rate, they had him on. He was playing a bicycle.
Johnny Carson.
No, before that. Who's the guy? Smack, smack. Who is that? It'll
I
come.
don't know.
I don't remember.
Yeah, you're too young, Alan. Yeah.
Well, no,
Jack
I listened.
Parr.
I listened to
What?
Zappa in college.
No,
Jack-
not
Jack-
Jack Parr.
Not Jack Parr.
No, we a bunch of friends of mine were into Frank Zappa in college. That's yeah, he was brilliant.
Alan, can you take the next one, please?
Sure. James writes, hello, Vincent et al. Shortly after posting this, an email from Stat containing, among other articles, one by Branswell, provides a link about H5N1 bird flu immunity was boosted by previous influenza A infection. I haven't read this article yet, but quoting from it, I guess, new study advances theory on why most U.S. bird flu cases have so far been mild. Researchers believe immunity to an earlier virus may play a role, but not everyone agrees. Indeed, count me in as one of the skeptics, says James. Yoshihiro Kawaoka, a leading influenza scientist at the University of Wisconsin-Madison, is among those who are skeptical, pointing to H5N1 infections in Cambodia, which, as reported, gives a link, 12 of which have been fatal. The version of this virus circulating in that country is different from the one that's been infecting cows and poultry in the United States. Kawaoka believes a number of other factors may explain differences in the severity of cases, including differences in the viruses, the way dairy workers and poultry cullers are being infected, often it seems with virus entering their eyes, or the ages and underlying health of the people who are being infected. I've been making notes in my posts that the eyes have the avian alpha 2/3 linked sialic acid receptors, as you pointed out. The upper respiratory system is almost all the human alpha 2/6 linked sialic acid receptors, sort of self-limiting infection. Further confirmed cases were treated with antiviral oseltamivir. I believe this was spread by contact, milk-snatching flies being one culprit. Richard Webby, another one I respect, had this to say, quote, "I don't want to downplay the study because it's an important study, but it's just explaining a part of the puzzle. It's absolutely not explaining everything we're seeing," he said. Continuing the quote, "We know that seasonal influenza viruses transmit just fine in the human population where there's a lot of pre-existing immunity. So pre-existing immunity in its own right is not enough to prevent an influenza virus from transmitting through the population. Still, if it makes the transition to human receptor binding domain, hope for the best. Would like to hear from TWiV or Immune about this." Yeah, I think we don't know why this has been milder. The route of infection is certainly a leading candidate in my mind, and all these other factors are potentially contributing.
The
Well,
late
Brianne.
night show host
Sorry.
that I was thinking of is Steve Allen.
Steve Allen,
Ah.
right. Funny guy. Brianne.
Ted writes, "Hi TWiV, many thanks for keeping us all informed. I'm a regular listener to the Saturday show and occasionally others. Recently contracted COVID as I began to stay at our cabin with an outhouse. Any concerns about the virus survival in contained outhouse waste? The outhouse was recently serviced. I also started Paxlovid on day 3. Will Paxlovid antiviral negate any waste issue? Best to add anything. Also, while I fortunately had an unexpired series of Paxlovid, I was shocked at the price increase to 1K to 1.5. This will make the best antiviral inaccessible to most. Sad. Best, Ted."
I think you're going to drink some bleach, no?
Yeah.
No, I'm just joking. Don't do that.
I
You
don't know
should drop
how.
some hydroxychloroquine into the outhouse.
Yeah, how are outhouses normally serviced? And I'm guessing there's probably some decontamination going on there.
Yeah.
There's
I wouldn't
also a
worry
lot of
about-
aerosol going on.
Yes.
I wouldn't worry about fecal transmission of SARS-CoV-2. It's really, there's no infectivity there. Sorry.
Yeah, and if your COVID symptoms started as you began your stay, I think you probably picked it up someplace else and brought it to the cabin, and it's
Yeah.
now, by the time somebody else stays there, it will have dissipated.
I'm just worried about other things in the outhouse before this.
I mean, it's an outhouse, but
Sewers are better.
Yeah.
well, right.
let's-
But if it's a cabin where you don't
I
have
understand.
that,
I understand. All right, let's do some picks of the week. Brianne, what do you have for us?
I have a recent XKCD post that made me laugh. It's called Geologic Periods and has a list of all of the geologic periods with a table that then lists my favorite part and my biggest complaint about each period. And this just made me laugh.
yes.
I liked, what were possible favorite parts during some of these exciting things like life developed or Earth might have had rings, or for the current period, burrito invented. But then
Love it.
also my biggest complaint, things like, evolution could stand to calm down a little, or newfound mold problem on Earth, or bugs too big.
I liked Google the Great Dying.
Yeah, Google the Great Dying. I also liked the complaint about the Quaternary period, which is the one we're
Yes.
currently in, was whoever picked this name for the third period of the Cenozoic.
Yes.
I like the Cretaceous. The good thing is
Yes.
raptors, and the bad thing is raptors.
That thing is raptors. Yes.
I like Cambrian. Evolution could stand to calm down a
Yeah.
little.
Yeah.
That's really good.
Very good.
Nice. Thank you. Rich, what do you have for us?
A recent sci-fi book that was introduced to me by a new friend. It's called Old Man's War by John Scalzi. And just very briefly, this was I'm a modest sci-fi fan. I'm not extensively read, but this was a new one to me. The deal is, it's way in the future. Humans have colonized the galaxy. That puts them up against aliens, so they need a colony defense force which you can sign up for when you're 75 years old. Okay. And when you sign up and you enter the Colony Defense Force, you have committed to leaving the Earth and never coming back. So nobody on Earth ever sees these people again. You don't really know what you're getting into because they don't really tell you, except that there is an understanding that your lifespan is going to be increased. You're even going to be made younger. Okay. And so this follows a guy who signs up for this and joins the Colony Defense Force. And I won't say anything more because there's too many spoilers. So it's pretty good.
Should I sign up? No, I want to do TWiV. I don't
Yeah.
want to sign up. Screw that.
And Rich, since you enjoyed that, you should check out Scalzi's Interdependency series.
Okay.
The first book in it is The Collapsing Empire. And then continue from there. It is a wonderful ride. It's just
Yeah, well,
great.
apparently there's 7 books in this series. It started out as a trilogy and then it keeps going on.
Right. So Old Man's War is he can keep adding to it. One nice thing about the Interdependency is that it's a trilogy and it wraps up, and it is
Okay.
absolutely the last book is like things end.
Okay. I will check it out. Thank you.
Alan, what do you have for us?
Yeah, so I saw Rich's pick. I said, okay, I got to pick this week. This is you could classify it as fantasy or you could classify it as sci-fi. It is a trilogy by N.K. Jemisin called the Broken Earth Trilogy. The first book in it is called The Fifth Season. It takes place on a far, far future Earth where civilization is not quite up to what it was before, because every few decades to few hundred years, there's a massive geological disaster of some kind. And people are, killed, and a huge disruption, and that's called a fifth season. Occasionally there's a fifth season, and they have laws that go into effect when there's a fifth season. And so these sort of fortified communities scattered around the map all hunker down when this happens. And this trilogy is about why that's going on. And it's just, I don't want to spoil too much, but there's a lot of geology in it that's really cool. And there's a class of people who are born with an ability to manipulate geology. So you would think these would be like all powerful wizards who are honored throughout the land. They are in fact a despised underclass. Which is a neat twist on an old trope, and the book just the world building here and the characterization, and when you think you know what's coming, just wait and see. It is an absolutely brilliant masterpiece of speculative fiction. That I think it is the only trilogy where all 3 volumes have won Hugo Awards.
Wow.
Yes, it is.
It is really, really amazing.
Yeah, I think the last time I was on an episode with Alan, you picked one of my other favorite books, Project Hail Mary.
Yeah.
And I laughed as soon as I saw this because particularly the 5th season, I like the 5th season the best out of this trilogy, is another one that I always recommend to people, and that is just absolutely fabulous. N.K. Jemisin herself is wonderful.
She can do no wrong. Yeah.
And so I second Alan's pick.
And even if you're not into sci-fi fantasy, just on a literary level, this is a great work of literature. I recommended it to my wife, who's not a sci-fi fan, and she was like, "Yeah, that was really good."
Yeah, I've recommended it to quite a few people.
Yeah.
All right, thank you, Alan. My pick is a short article in the BBC by Sofia Bettiza. Cacio e pepe, good food pasta recipe, sparks fury in Italy. So cacio e pepe is a great pasta dish, all right? It is made with pasta, pepper, and pecorino, and it's really hard to make properly, but it's delicious. And apparently this UK good food website published a recipe that got the Italians up in arms because it included butter. Oh, my God.
And they suggested you could also put cream on it.
It's cream, and these Italians are going nuts. You could read this. It became an international incident. They said, if you want to make a different dish, give it a different name. But cacio e pepe is just 3 ingredients. So said, "You have to yield to Caesar that which is Caesar's."
I love that because this is a Roman dish. Yes.
Yes, it's another kind of pasta. It's not cacio e pepe. Anyway, I love cacio e pepe. That's why um, I picked this. You should not make it with cream ever, but you have to get the cheese to melt. The Parmesan, not Parmesan, Pecorino, I think. Right? Let
Yeah.
me see here.
Pecorino.
And it has to melt and make it nice and smooth, and otherwise it will clot and be horrible. So it's a little bit tricky. And then in the UK, say you can make this a quick lunch. It's not a quick lunch. It's difficult to make. So it's very funny how the Italians got all bent out of shape. But after all, it is food, which they love, right?
And I
Which
love
we
how
love.
in this article, since it's on the BBC, they're taking pains to point out that the Good Food website used to be owned by the BBC, but is not anymore.
Yes, that's funny. We have a listener pick from Linda. For those who are Star Trek fans, the recent news article in Science will be of interest. And this is cool. This is DNA captured from the air could track wildlife, invasive species, and humans. And Linda goes on. It describes a device that samples the air, collects the DNA, sequences it, and uses this information to identify what life forms were in the environment. Star Trek fans will remember this is what the tricorder could do. It would sample the air and tell you what life forms existed on the planet. An avid listener, Linda. Tricorder could also do a lot of other things.
Oh,
It
yeah.
could
Tricorder.
diagnose your illnesses. Right?
Exactly. Tricorder
They
can
do
do
everything. Couldn't play back music, though, I think.
It's life, but not as we know it. Yeah.
All right, that is TWiV 1243. You can find the show notes at microbe.tv/TWiV. We can get your questions, comments, picks of the week, TWiV at microbe.tv. And if you enjoy these programs, please support them, microbe.tv/contribute. Alan Dove is at alandove.com, turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
Brianne Barker is at Drew University, BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time. I enjoy this.
I'm Vincent Racaniello. You can find me at Microbe TV on Blue Sky, Microbe TV without a dot. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.
The inmates are running the asylum.
Yes.
Mm-hmm.